Polymerase chain reaction, or PCR, is an efficient and cost-effective way to copy or amplify small segments of DNA. This allows clinicians to diagnose infectious disease, detect genetic variations and mutations, or track down the source of a viral infection, all from the DNA or RNA contained in a single cell or patient sample, such as blood, skin cells, saliva, or hair. PCR is accomplished in 3 distinct steps. The sample is first heated so the DNA separates into 2 pieces of single-stranded DNA through a process called denaturation. The heat physically breaks the chemical bonds between the double-stranded DNA helix. Next, primers are added in a step called annealing. These short, synthesized sequences of single-stranded DNA are used to mark off the selected segment of DNA sequence to be studied. Primers vary by diagnostic. For example, a primer in an HIV-1 diagnostic is different from a primer in a hepatitis C diagnostic. During the final step called extension, a heat-stable enzyme called Taq polymerase is used to fill in the gaps of each of the single-stranded DNA fragments at 70 degrees Celsius. This creates 2 copies of the original double-stranded DNA, with each of the new molecules containing 1 old and 1 new strand of DNA. PCR is repeated using the new duplicates, with each duplicate creating 2 new copies. "30 or 40 PCR cycles lead to more than 1 billion copies of the original DNA segment. Every synthesized DNA sequence of 1 PCR cycle becomes a template for the next PCR cycle, creating a chain reaction and exponential growth of DNA. The entire cycling process of PCR is automated and can be completed in just a few hours. In a healthcare setting, PCR enables a test that provide clinicians and other healthcare providers with information to guide treatment.